Hello everyone. Translational Biology Group, Department of Zoology, University of Calcutta is delighted to begin a new webinar series to celebrate the latest advances in the life science along with the discussing its relatedness to other disciplines called ant biology. In this new webinar series, we shall have fortnightly lectures on the weekend, Sundays for lectures on the different branches of life science, and Saturdays for talks elucidating other interesting facets of biology seen from a completely different perspective by exponents of other disciplines. In this way, ant biology aims to initiate discussions on cross-disciplinary discourses on various themes and the translational aspects thereof. We sincerely hope that you will enjoy participating in the talks and enriching this platform with crosstalks between apparently disparate but empirically connected academic disciplines. Today is our third lecture in this new webinar series. I am Payal. I will be your moderator for today's session. So, before we begin, I would like to remind you that you can post your questions in the YouTube chat box and we shall moderate that at the end of the lecture. You are free to interact with each other and with the speaker. And if you have any questions or comments for the speaker or for us, feel free to email us and we shall address it to the best of our abilities. And the feedback link will be provided in the chat box near the end of the session. And one more thing, please subscribe our YouTube channel. Our previous talks are archived in our YouTube channels. You can check them out there. Now, I will give you a brief introduction of today's speaker. Today, our speaker, Dr. Supriyo Das, was awarded PhD in biogeochemistry by Stockholm University in 2007. After finishing PhD, Dr. Das continued his research as a postdoctoral researcher at Netherlands Institute for Sea Research, University of Glasgow, Orebro University, and Linnaeus University. During his research, he received prestigious European Association of Organic Geochemists Scholarship, British Council, and Swedish Research Council fellowships. He is presently working as an assistant professor at Presidency University in Kolkata. His research applies elemental, molecular, and stable isotope geochemistry to understand the interaction between continually changing climate, environment, and human activities. He was a member of IODP Expedition 354 and 38 Indian Scientific Expedition to Antarctica. So, sir, without further delay, I would request you to please continue with your talk. Over to you, sir.
Thank you very much, Payal. Am I audible now?
Yes, sir, you are audible.
Good evening to everybody, and a special thank to Professor N.R.A. Banerjee for giving me this platform here. Before I start, I actually would like to have your kind permission. I mean, the audience, I'm asking. I want to have my sunglasses on due to my, issue with my right eye. So I think I will not look very weird, but please allow me to have my sunglasses on. So let me share the PowerPoint. So, hello, uh, is that
Yes, sir.
okay?
Yes, sir. All okay.
All right. So as I was introduced, I am a geologist, which is true, but I'll try my best not to have any geology or hard rock component in my talk. Uh, I'll try to make my presentation interesting and enjoyable for biology and geology students. Let me try. So I was a member of the 38 Indian expedition to Antarctica. That was in 2018, 2019. I was in Antarctica for a little over 2 months, and it was very exciting. I will share a video, the end of my talk. But there are many surprises. Of course, it is obvious because it's a different continent. And whenever we talk about Antarctica, we think of penguins and many other things. But one thing that probably we don't really think of is the sunscreen lotion. In Kolkata or many other parts of India, of course, in Australia, South Africa, many places, people do apply sunscreens before going out, going to beach for sunbath and everything. Uh, but in Antarctica, it was not a choice. Uh, we are supposed to have sunscreen before we go out. And in the picture, my face, you see that there are a lot of sunscreen. Now, it could be debated whether that much of sunscreen we need. We don't. I was like a sort of like paranoid when I just reached Antarctica, but we need to have it. And the reason we need to have it is something that triggered this research that I'm going to explain today to you. So when I went to Antarctica, I wanted to see how this climate change, ongoing climate change, leaving its record in geochemical archives like lake sediment or soil and all these things. But after I landed in Antarctica, something additional came in my mind, and that is what I'm uh, showing here. Now, when I talk about UV radiation or sunscreen lotions, most of you, I'm pretty sure, you know about the ozone hole in Antarctica. How it happened, this is also well known, I hope. And the reason for Antarctic ozone hole is mostly natural. That's a meteorological reason. And of course, there are some human component by releasing the ozone uh, destroying compounds in the atmosphere, but it is still there. Now, in 2020, sometime, the ozone hole is reportedly closed, but when I was in Antarctica, it was wide open and we had a threat. One thing we should remember that this ozone hole is not a very old phenomena. It started like probably past 100 years, noticed in 1980s, a little bit earlier than that. So whatever happened in between in last couple of decades was not something that happened before that. So it was something uh, new for the vegetation for life in Antarctica. So they need to adapt the changes as we do. But when we go, we use sunscreen lotion. Uh, the vegetation over there may not be able to use that. So we'll see how it happens. Uh, I I was stationed at Bharati Research Station in Lastman Hills. That was in East Antarctica. Now India has two stations. One is Bharati, another one is Maitri. The first Indian station, that was Gangotri, went under the glacier. It was in 1983, 82, something like that. And then the next station came up, which is Maitri, started operating from 1989. And this one, the Bharati, is pretty new. So it started operating from 2012. It's very new and excellent facility. You see, it looks fabulous from outside. It is very interesting inside as well. It looks like a three-star hotel, even five-star you could say because it has a helipad. But anyway, when we talk about Antarctica, we think of cold, right? It's like extreme cold. In Lashman Hills, it is not like that. When we landed over there, we found it very warm. It was extremely warm. Literally, we could go out from the station in t-shirt and shorts. We are not allowed to do that. The reason we are not allowed to do that because the climate uh, it's extreme. It's called extreme not because of a certain moment of time. It can change. The weather can change very fast. And if it becomes windy, even the temperature probably +2, you will feel -20 uh, at some point, and you will die. Very soon. So the climate is harsh. The climate is very notorious. So you have to be very careful when you are going out. But anyway, Lastman Hills is very warm when I went there, and it is not only for that particular time. It is the warmest areas in Antarctica. So the summer temperature becomes very high. I when I was there, it was +2. The average is about zero point 0.6 and something. The coolest time we can find -15, minus even a little bit more. I mean, it can go up to -30 to 40. But I was not there during the winter. Uh, Another important thing about Lastman Hill is that it's an ice-free zone. So when we talk about or think about Antarctica, we think of white continental ice sheets. It is there, but not in Lastman Hills. Of course, in winter, snow accumulates and it becomes white, of course. But from early spring to summer, the snow melts. So if when I went there in summer, uh, little snow was there. It is mostly barren rock. One thing that we also remember that Antarctica do not have any rainfall. So whatever water is available, the fresh water I mean, that is only from the snow melt water. And the meltwater accumulates in small depressions. It may form ponds, small lakes, and there are about or over 150 lakes and ponds in Larsen Hill areas. The life, if we talk about life in Antarctica, of course, we think of penguins. They're there, but apart from penguins, there are birds, squaw, for example, and there are a lot of moss and algal mats. The algal mats, you will find the algal mats under the lakes, large lakes. It could be at the bottom of the lakes or around the fringes. But uh, when you look for it, you have to be very careful because you'll also find moss in the same place. So moss you will mostly find in the snow melt area where the channels where the meltwater flows into lake or into the sea, and algae you will find uh, in water areas where water accumulates. In this picture on my left, you'll find squaw. They are predators. We are not supposed to be close to them or feed them, but that's a different story. Now, I mean, many things I noticed after landing in Antarctica, in Larsemann Hills especially, and one interesting thing that I found is the change of color of the moss. So some moss beds they are green, few are red or greenish red, and the farther I go from south to north, the
Sorry, I think there are some technical issues. Please wait.
and with good immunogenic response against COVID-19 disease. So trust the vaccines made in India. Do take the vaccine at your turn and do not believe in hearsay. Remember, dawai bhi aur kadai bhi. Even after taking the vaccine, continue to wear a mask, observe physical distance of 2 meters, and regularly wash hands. For more information, visit www.mohfw.gov.
We will soon, Sir will soon join us. He's having a technical issue and our moderator is contacting him. So, for our viewers, please wait for a little bit. We will soon continue the program. Thank you. Yes, sir.
Yeah,
Sir,
I
please
don't know.
share your PowerPoint again.
So when I was disconnected, do you know? I mean, from which slide? Can you tell me?
Yes, sir. Your fourth fourth slide.
Okay, let me do this. So I was disconnected when talking about this slide, right? This this one or this
Sir,
one?
this one, sir.
So okay, so this was the last slide I talked about, right?
Yes, sir.
Okay, I'm sorry. So I think some of you tried to call me, but I kept my phone mute. I'm sorry. Now, anyway, so what I start from here. So uh, I was stationed at Larsman Hills, and the research station is known as Bharati, right? And when we talk about Larsman Hills or when we talk about Antarctica, we think of white continental scenes, which I say that it is not. It is there, and you can see from Larsman Hills, but Larsman Hills is mostly famous for its ice-free areas. That means during the winter, the snow accumulates, but in spring and summer, the snow melts. And when I went there, I found barren areas like rocks, lot of rocks, little snow left, and so on. And in Antarctica, the only source of water, fresh water, I mean, is snowfall. There is no rainfall. So that's what I said. So from here, I think I disappeared. So in these pictures, you see that small ponds and lakes So there are about 150 or more than 150 lakes in Lashman Hills, and those lakes, those are freshwater lakes, and they form during the summer or in early spring from the meltwater, and they support the vegetation and little bit life, whatever is there in Lashman Hills. Of course, when we talk about life, we think of penguins, but there are other birds as well, like skua. You can see skua right over here on the left picture. Now, these lakes are oligotrophic. That means they're pretty clear. There is no phytoplankton present in the water column, but there are benthic algal mats that resides at the bottom of the lake or around the fringes. Uh, is that okay? I'm still there, right?
Yes, sir. Yes, sir.
Okay.
All are okay.
I became paranoid. I'm sorry. So there are other form of vegetation that is moss. We find algal mats, which is in the moist areas or mostly in under the water, and then the algal moss. Sorry, in the moss beds. Now, the first thing that I observed in Lusman Hills is a change of color of the moss beds. Uh, some moss are green, some are red, reddish green, some are gray, black, or even completely dry. The change of color is not something that I observed first time. It is well noted, and people did some and researchers did a lot of research on it, and they found that it is mostly due to the water stress. Like moss, they grow at the moist areas, but with time, the water dries up and they become stressed. So the green moss indicate there's a lot of water, and when it become dry or gray, and they indicate there is a water stress. That's how people understood. Now there's another component which is the UVB radiation. The moss they mostly stay at the open areas, so they receive a lot of sunlight and UV radiation, and they also protect themselves from the UV radiation by changing the color. Now, what I observed in Lashman Hills is that the green moss mostly resides in the shades. That means now in Antarctica, in summer it's 24-hour sunlight, so there is no time when you can say sun sets down or something like that. So it is sunlight throughout the day, 24 hours, and there are certain areas where there are shades. So the green moss, I observed them in the shades mostly, at least for part of the day. They don't have to face the sunlight UV radiation. And the moss beds, which are in the exposed areas and in dry areas, like the meltwater channels which dried up, they're black. They are called moribund moss, and they are like blankets. Somebody threw a blanket on the ground. They're thick. They are like few inches, like 4 to 5 inches thick, and they look like folded on the ground. The picture you are seeing here, these are dry moss, dry moribund moss, and the whitish part on the surface are lichens. Now, what we knew about moss and their color changes is that, as I said, they indicate water stress, and they also help themselves to protect from UVB radiation. Uh, and the compound they produce to protect themselves from UVB radiation is called UV screening compound. The composition of the compound is not very well known or well understood, and that differs from different moss species and from different areas where the moss is growing. That also have some control on the screening compounds. Now, the problem that I felt is that why we don't know the composition of the UV screening compounds in the mouse. The problem that may be there, or I understood, is that the biologist or the botanist who actually study the moss, they are not biochemists or biogeochemists, so they look for compounds which they felt could be the reason, but it could be something else. So having a geology or you know, I don't say a geologist myself anymore, biogeochemistry background that I have, I actually thought of why not alkane. Now, if you look around, you find any plants, uh, you will find alkane in there. You know, they are the most basic composition of plants, and we look for alkanes even like in geological record. So they are everywhere, and most they also have alkanes. Of course, they do. So what role the alkanes may play in those mosses? So that was something that came in my mind when I say uh, alkanes. You will find alkanes in all plants. That means that if you say if you look at a waxy surface of a leaf, the waxy part that contains alkane. Now that is also, I mean, you know better than me, epicuticular wax, and their function is different than extracuticular wax that you have in mosses, but not very different. They work quite in a similar way. So moss also produce alkenes for almost the same reason, and we didn't study them. That is what I wanted to say. Uh, Another important thing that I must discuss here is that the benthic algal mat, which is living under the water, like in a deep lake. The lakes could be deeper, 30 meters, or could be shallow, but the algal mat that is residing at the deep part are green. They become reddish green at the shallower part, red, more shallow part, and white gray, if not black, when they come to the surface of the, I mean, at the fringes of the lake, where the what you call this film is very thin. So they also change color. But now, if we consider UVB radiation as the reason for the color change for the algal mat, that has no influence of water stress because they are living underwater. So water is plenty over there, and if they have any problem, that is from the UV. Now, another important thing that we should also remember here that the water is very clear. They are oligotrophic lakes, I mentioned, right? So the water column may not protect them from UV radiation. Although most of the time you'll find the lakes have a thin ice on it and a lot of snow on the ice, so probably it helps it provides some protection to the algal mat, benthic algal mat, but it is not a huge protection. But of course, like something is exposed to the sunlight and something is under the ice and snow cover, have some effect. I think some difference there should be. So that is what I understood from the field observation. So what I did, I collected a lot of samples. The Lashman Hills, it's the part in the map is Lashman Hills, and our Bharati station is right over here in the Gulf Peninsula. So all this it is close to 13, point number 13. So all these dots and points that I mentioned there that I have on this map, I collected samples from there. Uh, when the sea was frozen, I used a snow scooter. When the sea melted, I had the privilege to have a helicopter taking me here and there and help me collecting the samples. Uh, so I analyzed N alkenes. N stands for normal alkenes in the moss and benthic algal mats. Now, uh, you may or may not be familiar with alkenes, so let me introduce it. These are like very simple straight chain saturated, we call hydrocarbons. That is hydrogen and carbon. They have, and they could be a short chain. That means the carbon number could be less from, like, you have this the gas what do you call the cooking gas that we use in our kitchen, which is very expensive these days. They also have some alkenes in gas form, but you won't find those small molecules in plants. They're a little bit larger, but not very large. So here the C17 to 20 that actually indicate the number of carbon atoms in the alkane molecule, right? And C21 to C30 that also indicate the number of carbons in the alkane molecule. So we call it short chain when it is less than 20 carbon molecules, 20 carbon atoms in a molecule, and long chain when it is above 20 atoms in a molecule. So I divided the alkanes into groups and make a ratio. Now, what I found is that the ratio changes with the changes of color. If we take, for example, if we take the benthic algal mass, you know, the green mat, they produce less amount of short chain in alkanes. The more it goes towards gray, like from green to reddish green to red and to gray, they produce more and more short chain in alkanes. Now, as I said, these algal mass they have very little to do with the water stress because they are living underwater. So, if they are changing their color, that has to be connected with the UV radiation or increasing UV radiation because the green mat they lives at certain depth where probably UV radiation reaches but in less amount, and the shallower they become they change their color from green to red and to gray, and with this change of color they produce more and more short chain in alkanes. What happens in moss? That is little bit complicated. Complication comes because of the water stress. Now green moss, they live in shades. They don't have water stress. They don't have much influence of the UV. They do have because part of the day they are exposed to the sunlight, but sometimes they also live in the shade. The grey moss, they are exposed to sunlight. They have water stress, but they are still living in the moist areas. The moribund moss, they are almost dry. I mean, the water is limited. They are not dry, but they have some water stress. And dry moss, they are also moribund moss. They have extreme water stress. Now, if I compare green moss with grey moss, I find that grey moss produce more, little bit higher amount of short chain inalkanes in compared to green moss. But in moribund and dry moss, the short chain inalkane component is very less. The reason is that if we consider the short chain inalkanes are, I mean, the production of short chain inalkanes are directly related to the UVB radiation, then gray moss is the best candidate, an unfortunate candidate that receives a lot of UV radiation. Moribund and dry moss, they also receive UV radiation, but they have bigger problem. They have less water, so they had to survive in very little water, or they what they do, they actually desiccate themselves. Okay, they protect themselves in a way that the less water or water stress doesn't harm them much. So they produce long chain inalkanes that keeps them intact. They don't break down, they don't disintegrate or die. So in a very simple term, if we consider the UVB radiation is a major you know controlling factor of the change in color of algal mat and terrestrial moss, we find the more the vegetation is exposed to the UVB radiation, they produce more sort of in alkanes. They change their color at the same time. The color may not have anything to do with the alkane, but the alkane has something to do with the UVB radiation. Moss does the same thing, but they have additional constraint of the water stress. So both combined, inalkanes has very important role that they play in Antarctic moss and benthic algae. So in conclusion, I will conclude here as well. Uh, this is something new that we observed in Antarctic vegetation. The inalkane composition in Antarctic moss and benthic algae, they reflect the amount of UVB dose they receive. And the important part is that it is not something they're having for last several thousand years. They adapted the change in very recent time because the ozone hole didn't happen early Holocene. It happened in this, I mean, in last century, right? So they actually adapted something, and that is new. So they don't have UV screen. Yeah, I mean what I call the sunscreen lotion that we use. They adapted something. They changed their molecular structure. They produced something, some molecules that help them to protect themselves from the UVB radiation. So that is something I hope is right. We submitted a manuscript to Nature Communications. We'll see how it happens with that. But I will conclude here. I will not extend. I mean, I will not torture you anymore. So before I say thank you, I should acknowledge NCPOR who actually took me from Kolkata to Antarctica, and all logistic personnel, our leader P. Lango, who helped me with sample preparation, my students at Presidency, Prabhat, Subham, and Madhurima, and my colleagues, Arindam from BSIP, Priyank from Presidency, and Devanita from ISC. These are all references. If you need to know or if you need to read any of this, please let me know. I will send you the reference. And thank you for listening. Uh, Now I will share a video, if you allow me. So I guess I should stop sharing my screen. All right. Hope everything is well. You could watch
Thank you,
and
sir. Thank you for the wonderful video and very informative and innovative talk, sir. Thank you so much. Sir, we have few questions. Should we go
Mhm.
ahead with the questions, sir?
Yeah, yeah, yes, please.
Questions from Shahili Bishwas. Has
Mm.
COVID pandemic in different countries put a positive effect on ozone hole in Antarctica with respect to pollution?
That that could be a reason, but it is a long-term effect. If you uh, uh, consider the reason for having this fall, uh, it is mostly the meteorological effect. The way Antarctic stratosphere to become isolated from the rest of the part of the world is a primary reason. So I'm not the right person, I think, who could make a comment, but definitely it may have some effect. Because I don't know the residence time of the ozone depleting compounds that present in the stratosphere and how they actually work. So if it is a short time span that has this effect, and probably the COVID-related lockdown has some impact. But I don't find any literature explaining that right now. So probably in next few years it will come out.
Okay. Thank you, sir. Uh, Akashinda Marik wants to know, are similar kinds of alkanes present in algae other than Antarctica?
Yes, alkanes are present in all sort of plants. Okay, so the short chain alkanes that is also present in any algae. They could be a little bit shorter, a little bit longer, but the same kind of alkanes are present in algae elsewhere.
Okay. Thank you, sir. And Akashinda also wants to know that are these alkane ratios or compositions also get affected in areas common for oil spills or water pollution? Can we use this as biomarkers for UV radiation amounts?
Uh, okay, I didn't say much about alkenes during my talk because I didn't know how the audience will, you know, like it or not. Uh, Two things. First is that alkenes are present in hydrocarbon, in petroleum hydrocarbon, I mean. They are present in vegetation, they are present in soil, uh, even rocks. So they're everywhere. And they are primarily produced by vegetation. Okay. The source is vegetation. They get degraded, they get decomposed by bacteria. So their signature, I mean, the ultimate signature that you may find in water or in sediment or any geological archive like rocks or something that has gone through a lot of things before they actually came to that place. What I mean is that, say for example, if you are looking into in a water sample and it has alkane, definitely it will have alkane. It has particular organic matter, right? Particulate matter, and that particulate matter may came from the seagrass, if it is seawater sample, for example, or if it is a river or lake water sample, then it may come from vegetation in the catchment area. Now, think of a leaf, yeah, fall from a tree, and coming to the rainwater or river water and coming to the lake. The leaf will not remain intact. It will be attacked by the bacteria. They will chew the leaf. It will break down through the physical force or other forces. So what you will find in the water, either dissolved organic material, dissolved organic carbon, you could say, or the particulate organic matter, and they contain alkanes. Now, the actual composition of alkane of the leaf and the composition of the alkane you will find in the water, that in the form of dissolved organic carbon or particulate organic matter, are not the same. You have to understand the processes that transported that alkane, the processes, the biological and physical or chemical, biochemical, whatever process that alter the alkene molecule. Now, the problem is that if you want to isolate one process from another, you need to know the actual transport mechanism, the influence of all these individual processes separately, and that is very complicated in nature. I mean, when I am talking about the UVB radiation in Antarctica, I know there is little pollution. I know the other factors that may influence my alkene signature is either water stress, drought conditions, or the species of the moss that I am analyzing, or I don't know if, but probably few. I mean, two or three conditions that may influence the alkane composition in my sample. When you are studying alkane uh, in a lake or in Ganga water, for example, if you are doing that, there are lots of conditions that we don't know. So unless you know all these conditions, you cannot. I mean, it is very difficult to isolate the effect of UVB radiation on the plant composition and the alkene compound that you are studying. So I don't think it is a straightforward. But if you could find out a place where pollution is less, the other constraints, conditions that may alter the alkene compound, uh, I mean, the influence of other conditions are less, then you may find out if there is anything to do with the alkene composition and UVB radiation that may be done. But and at the same time, probably somebody needs to do laboratory scale analysis. Growing the plants under different UV doses and analyze the alkanes. That is the easier part that can be done if anybody wants to see whether UVB radiation has anything to do with the alkanes or not in other part of the world. I think I tried to explain what I understood about alganes and UVB radiation. Is
Yes,
that what you wanted to know? I mean, Avilina, I mean, I can read small letters. What is
Akashlila.
Akaslina? Okay, sorry. I'm sorry. I cannot see
Akashlila.
small letters. Akaslina. I cannot see small letters. Is that what
Okay.
you wanted to know?
Thank you, sir. See, can we use this as biomarker for UV radiation amounts?
Uh, we could, if we know the other conditions that can alter the alkane composition in the archive. Like if you when you are saying that whether you could use it as a biomarker, to me it is about the past changes, right? In a geological archive. So for that, you need to know other conditions that can alter the alkene compounds. That's what I said. Like you don't know in natural conditions, there are a lot of influences that can, that can alter alkanes. Alkanes are very common, and they can be degraded very easily. And what I mean, if we have a long chain alkane, for example, or a short chain alkane, the ultimate product will not remain the same. They'll be different. They'll be branched alkane could be, or they'll be saturated from branching. So there are a lot of changes the alkane will go through during the transport and deposition and diagenesis. So all these things need to be known before we could use it as a biomarker. We could say that this is exactly what is happening or what happened with the alkene molecule. I don't think it has any future as a biomarker for UVB in near future. We need to know a lot about it. Unless we know that, we cannot use it as a biomarker.
Okay. Thank you, sir. Thank you for answering the questions. Sir, that's all the questions
Okay.
that we have today. Thank you so much, sir.
Thank you.
Thank you.
Thank you very much. Good night.
Thank you, sir. Thank you, everyone. Uh, Thank you. Please subscribe our YouTube channel. Our previous talks are archived in our YouTube channel, and you can check them out there. And please join us tomorrow again. And thank you again for joining us today. Take care. Good night.